Photobiomodulation. We can't live without photobiomodulation. We really depend on the sun, etc. We can't live without it. Okay. Now, what is it? Basically, it's non-ionizing light source, and it's non-thermal. Okay. And basically, it involves low-intensity light, basically modulating cellular functions. So think of your body, your organs and cells as a computer hardware, and the pathways as a software. And we're attempting to, to basically influence these pathways, these software. Now, this is a slide that basically sums up my entire lecture from start to finish. So we can see here photomodulation wavelengths. What is its targets? Well, its targets are lipids in the membrane. Its targets are water. Its targets are mitochondria, heme groups, and also some cytokines and things like that, affecting pathways, eventually going ahead and giving you these things down here, the various cell types. Okay, so basically a very important aspect of photobiomodulation is its effect on calcium signaling. Remember now, your cell membrane, which is the eyes and ears of the cell, lets things in and out of the cell, allows calcium to go in. And that's what really controls a lot of things, a lot of pathways and things like that. You can see it here on that illustration. All right, so basically the many phases of photobiomodulation, angiogenesis. Now remember, there's a difference between angiogenesis and vasculogenesis. Vasculogenesis means you have nothing there to start with, no vessels. Angiogenesis means you're building on vessels that are already there. So vasculogenesis, you need stem cells. Angiogenesis, you can get by with just PRP. Okay, so here we are here, the various types. Now, ionizing radiation, which is sort of these guys over here, basically kicks out electrons. It does a lot of damage to cells, and it can be detrimental. Now, sometimes they'll use radiation therapy in cancers and things like that, and that's why they're doing that. Okay, so we can see here the various types of electromagnetic radiation. And you can see here the various wavelengths, and that's old mnemonic Roy G. Biv to remember the different color lights and how they line up. All right, but we can see here how some of these colors penetrate. Ultraviolet doesn't really penetrate that much. Visible blue, and as we keep going further in the spectrum, we get the best penetration as infrared. Now, basically, I just wanted to go a little bit into laser versus ordinary light. You can see one is monochromatic, one is not, one is directional, one is not, one is coherent, one is not. I didn't really want to get into the aspect of lasers in this particular talk. Okay, so this is the physics, but I think the next slide shows it better. So we can see here some of the obvious things. So we have wavelength, which is right here, the distance between successive crests of a wave. You have the frequency, okay, meaning how many cycles pass in a given point, and that's extremely important. We'll get into that momentarily. Then we have the amplitude, which is basically, what the strength is more or less, and then we have the velocity. Now, duty and frequency, okay. Frequency, how many pulses per second, and the duty is, in that pulse, how much is that time, is that light on, okay. These are important concepts to kind of think about. Now, these are various frequencies that can have profound effects on the body. Look at this, healing of capillaries, healing of a herniated disc, fluid retention. You basically can pulse your light at these frequencies. Okay, this is getting to be in the new field of vibrational medicine, energy medicine, things like that, which I'm certainly not an expert in any way, shape, or form. Now, you have these things called the Noninger frequencies, okay? You can see that there's 7 things here, and they can do various things. Emotional reactions, coordination, and this sorts of get into the frequency. This is why we now realize that pulse light is so much more important than continuous. If you're using continuous light, it's going to work. Not going to work as good, though, by no means, okay? So you can see here, pulse light therapy, photobiomodulation. And you can get an idea about the pulse when you break a wine glass. Remember, they used to have a commercial years ago on TV. They had these opera singers, and they would get such a high pitch that they could actually rupture the glass. And that's what we're trying to do with some of these organisms. They have something called the mortal oscillatory rate, and you can see some of those here, like coronavirus. If you can get these wavelengths of light to basically be pulsed at these frequencies, you'll do some really good things. All right, so basically, why is pulse light more important? Well, basically, it can penetrate deeper into the tissue without causing as much damage because it's not on all the time, so it's not going to heat up things as much. So it has a much greater biological effect. And here's some actually scientific aspects. So when you basically have more of an on and off period, you can maximize ATP production. You basically minimize some of the reactive oxygen species. It can show that the pulse light of 10 frequencies can stimulate ATP production more. And you can see, you go on. And again, the slides are available to all you guys, so don't worry about that. So we get into this kind of mixture now of photobiomodulation and vibration medicine. And they basically, what is it? Well, you have energy resonance and cellular communications, mitochondria. Again, when all is said and done, most everything we're at this meeting comes down to the mitochondria and how they're healthy and things like that. So you have biophotonic and electromagnetic interactions and basically holistic effects. All right, so you can see here, so basically the similarities, photomodulation, mitochondria boost, mechanical transduction, systemic effects, circadian rhythms. These are all, this is that new field that's really coming into play now, okay? So now there's a new thing called biophotons, the new field of photoscience. We all give off light, which is amazing. I didn't realize that until I got down into this a little deeper, and there was just an article a couple months ago that says this, that says, you know, they were looking at the emissions from living and dead mice. Significant difference. This is not just in people or animals, it's throughout the entire plant and animal kingdom. Living things give off light, and they're going to use that more and more, I suspect, as time goes on. And you can see here, now we get to the field of optogenetics, where basically we're combining optics and genetics to start getting some interesting things that are going to happen. And shining light on target cells expresses opsins, which can precisely control their activity and basically stimulate growth factors, all sorts of things like that. So we get into this new field here, and then we can talk about the photoreceptors. So we have the opsins, which are photosensitive G proteins. We have the cryptochromes, which are flavoproteins. Remember that electron transport chain. And then we have phototropins. But this next slide shows it a little better. Okay, so you have your opsin group over here. What does it do again? It affects calcium signaling, which affects so many different pathways. Then you have flavoproteins, light activation, transcription of chromatin, again, gene expression, calcium and lipids, protein kinases. Remember, everything is built on proteins. Without your proteins functioning properly, you have trouble. Okay, then we have a chromophobe. So basically, it is a compound that absorbs light energy and it transfers it to another molecule, kicks it up, so to speak, as the old saying goes. And you can see that here. So you have a photon, and it gets excited, and it kicks that electron up, and it forms a hole, and then it becomes a whole complex called a cytron. Now we can see here about the chromophores, okay? We know one in particular is Cytochrome C Oxidase, but you can see here, for instance, green, again, starts affecting calcium. You have infrared, calcium, and then you have mitochondria, et cetera. And you can see here, so Cytochrome C Oxidase is the primary chromophore in the body, red and infrared light. It regulates reactive oxygen species, which are a very important signaling molecule. You know, how many reactive oxygen species you have can really dictate which pathway is going to be involved. Remember, you have two major pathways. You have the NRF2 pathway, which is our buddy. It's basically the major pathway to reduce inflammation in the body, regulates gene expression, hangs out in the cytoplasm till it gets kicked into the nucleus. It's a transcription factor. Remember, transcription factors transcribe the genes. They basically tell the genes, do this, this, and this. And then you have NF-kappa beta. Certainly nothing we like in regenerative medicine, but sometimes when we have an infection, it is our lifesaver because it can really get rid of those bacteria, viruses, etc. But it causes inflammation. Okay, so we can see here, harnessing light through the mitochondria. And basically what happens is we'll see cytochrome C oxidase, ATP synthase, and eventually what does it do? It makes ATP, the body's energy currency. All right, so we can see it here. We're kind of doing a cross-section of the mitochondria. Basically, we get a proton gradient. Remember now there's 5 parts of the mitochondria. Cytochrome C oxidase is in 4, but eventually 5 is where we get the magic that happens. It makes the ATP, and it's oxygen consumption. And we can see that here. Now we start getting into the electron transport chain more. And I used to think, I don't need to do this anymore. So now I left medical school, I said, that's it, never need it again. It's so important for everything that we do. But we can see here, here's cytochrome C oxidase right here. Okay, and it's the terminal enzyme complex in part 4 of the chain. And this is the chain right here. Now let's kind of go over things a little bit. Our buddy NAD right here, complex 1. CoQ10, complex 3. Cytochrome C Oxidase. Now, how is this important? The next slide shows you a lot of how it's important. Look at your neurodegenerative diseases. Alzheimer's, Parkinson's, Huntington's, ALS. All are related to the electron transport chain. Remember, these are neurodegenerative diseases. They're basically, the energetics in the cell is kaput, not working well. We're actually hopefully days away from the FDA giving us an okay for a ALS. We're using it with the hemolumin, and then we're going to put it on right on with the ozone. So I'm keeping my fingers crossed, but should be no problem, we think. All right, so let's take a look at some red light. So red light in the 600 range, well, what does it do? Well, we know cytochrome C oxidase. But when you're using infrared, 800 to 1064, it affects the cell membrane. Remember, the eyes and ears of the cell. It lets things in and out. But what does it do? It affects calcium signaling, which then affects cyclic AMP, reactive oxygen species. So this can help with differentiation and proliferation, something we really want to do when we're doing cell therapies. So that's the importance there. So that's why you want to use both infrared and red light if you're doing some cell therapies, okay? Now, production of nitric oxide from photobiomodulation. This is kind of a rough view, but we're going to get into it just a little more. So we can see here, you're going to start making some nitric oxide, and it's going to get dissociated from the cytochrome C oxidase, and eventually it starts doing a lot of other things. Nitric oxide is one of the most important gaseous signaling molecules in the body. There's 3 gaseous signaling molecules: carbon monoxide, hydrogen sulfide, and nitric oxide. But they're all important. We'll get to that shortly, okay? So photomodulation involves photodissociating. In other words, the light will make the nitric oxide leave the cytochrome C oxidase and can do other things in the body. Now, this is an important aspect here. Nitric oxide acts as a gatekeeper that makes the respiratory cycle run by controlling blood flow to tissues, a function more important than the absolute amount of oxygen carried by hemoglobin. Now, we just heard a talk about the hyperbaric oxygen. I came in late, unfortunately. I didn't realize he was talking. But one of the things to really I call nitric oxide the poor man's hyperbaric chamber. Because if you look at the original research by Dr. Tom from the University of Pennsylvania, he said, hey, the real basis of hyperbaric oxygen is the secretion of nitric oxide, which then stimulates the bone marrow, releasing matrix metalloproteases, increases stem cell output by 8 times over 20 sessions. So give your patients nitric oxide whenever you're doing any type of regenerative procedure. It's going to just make it work better. And if you're using hyperbaric thing, give them nitric oxide all the time. I basically use Neo 40. Works good. So here we see here. Now we can see here a very important aspect. There's, the hemoglobin. You have this A3, and that's what's binding the nitric oxide. All of a sudden, when you have photomodulation, it releases that nitric oxide. And this is what happens. Then you start getting more downstream signaling. You get activation of transcription factors, which are going to start transcribing messages with the genes, anti-inflammatory protein synthesis, anti-apoptotic, cell migration, cell metabolism, etc. So a lot of important things that go on there. Okay, now, basically we can see here. So what does nitric oxide do? Promotes, sorry about that. See if I can go back. It promotes mitochondrial growth, fine-tunes respiration, basically mitochondrial function, protects the cells, redox signaling, and controls mitochondrial shape and fusion. Now, one of the newest thinking in all the world of stem cells is the stem cells may really work on transferring mitochondria. It's not like, I'm going to have this cell and it's going to all of a sudden make muscle tissue, cartilage. It's transferring mitochondria to diseased cells that are there. And that's a very important concept. So you can see here now why we have that, how this is going to basically affect mitochondrial dynamics. Okay? So you can have a fusion of the mitochondria or you have fission. Fission is a way we kind of get rid of some of the diseased mitochondria, mitophagy. And one of the best things you could use for mitophagy is urolithin A, if you're familiar with that. If you're not, become familiar with it because it's very important for the health of all your patients. All right, so we can see here, so mitochondrial transfer is basically how it works. They basically form these tunnels, and they basically start transferring one mitochondria to another, and I think that's going to be. Remember now, a lot of your exosome products one of the components of exosomes are mitochondria. So that's why those exosomes can be beneficial, and that's why we can photoactivate some of the exosome products, because when it up those mitochondria, they work a little better. Okay, so basically though, not all is good with nitric oxide. Under certain circumstances, it can be detrimental. High concentrations can basically inhibit activity of mitochondrial respiratory chain. Excessive triggers mitochondrial permeability. You know, we were running into this with the COVID a number of years ago. Remember the cytokine storms and things like that? What was happening is, among other things, we were getting too much, basically, nitric oxide. So remember the hermetic effect, that's that bell-shaped curve. I believe I have a slide somewhere here. But anyway, this is where we see that. So we look here, low concentration, a lot of good things, okay? We get nitric oxide release, we get oxygen used, ATP, but high concentration, it basically starts blocking those, gives you cell death, stops differentiation, and does a lot of bad things to you, okay? Now the sirtuins, remember there's like 7, so I call them genes or whatever you want to do, enzymes, whatever, but basically can do a lot of interesting things, okay? And the photomodulation can really basically affect the sirtuin genes in a very positive way. Now these are the sirtuin genes that are involved with the mitochondria. Now basically in mitochondria you have SIRT3, SIRT4, and SIRT5 found in the mitochondria, but SIRT6 also, which is out in the cytoplasm, has a very profound effect on mitochondrial health and regulation. Okay, so here you are, the many different jobs of the sirtuins. And this is one of the things that we know NAD can be beneficial with and things like that. So look at all the things here. So everything here, if you get these all corrected, your patient's going to be better, okay? Now, it's been shown to increase and stabilize telomerase, which is a very important aspect of things. Remember, telomerase is that enzyme that puts the telomere end back on. And that's an important aspect of anti-aging and things like that. So we can see that here, something called the Shelton genes and things like that. But here it is right here. So you can see it, and it helps. And I was talking to Bill Andrews, a good friend of mine, who's probably one of the world's experts on telomerase and things, and he said this is really a good thing to do. All right, so basically photomodulation and hypoxia-inducible factor. Very important thing. That's another thing I have a lot of interest in, how controlled hypoxia is one of the best things one can do for their health. Now, intermittent hypoxia, you know, that you'd get with sleep apnea and things like that, or I should say continuous hypoxia is bad for you, but intermittent hypoxia is a good thing. Now, when you have the hypoxia-inducible factors, photomodulation basically will stimulate some reactive oxygen species against some oxygen, and what this does is it kicks these guys, which are in the cytoplasm, which is here, it goes into the nucleus, and it starts giving, again, it's a transcription factor, starts giving a lot of different information, increases angiogenesis, erythropoiesis, apoptosis, glucose metabolism, so cell proliferation and survival, a lot of interesting things. This is a very important transcription factor, and photomodulation turns it on. Now, here's an interesting thing, because the hypoxia-inducible factor will stimulate hemoxygenase 1. Okay, and what does that do, hemoxygenase 1? Well, it makes a couple of interesting things here. Okay, basically it makes biliverdin, which is a very potent antioxidant. It also makes bilirubin, and it also makes ferritin, and it makes carbon monoxide. You cannot live without carbon monoxide. But remember that hormesis, that bell curve, okay? Now, because of this, you know, there's some of you out there, just to kind of change gears a little bit, that say, oh, you can't have any red cells in a regenerative product because it's so bad for you. Absolutely baloney. We published a paper 4 months ago, very well-received patient, why RBCs in a PRP product aren't a problem. Just for this reason, because it basically makes RBCs become anti-inflammatory. And I'll give one example why RBCs are not the inflammatory thing that you do. If you have a spinal leak, what do? You put whole blood on top of the dura. If it was so inflammatory, would you do that to the dura? I don't think so. All right, so moving on. Photomodulation and stem cells. Well, that just basically common sense. It stimulates another number of different pathways. Anything that's going to essentially make more energy is going to also go ahead and make your stem cells better and basically make more of them, okay? So we can see that right here. And this is the various pathways. So it can help with cell proliferation, migration, growth factors, signaling pathways, reactive oxygen species, and modulation of pathways. All right. Again, this is just another slide showing you a little different. Again, transcription factors, epigenetic mechanisms, and signaling pathways, like the Wnt pathway, TGF, things like that. All right, and this is basically showing how certain colors like red and infrared can basically help with differentiation. You can get osteoblast, osteoclast, and things like that. So we think this may have an effect on osteoporosis, among other things. We have a mechanism where, or I should say a regimen in our office now, we're using hyperbaric oxygen, we're using some stem cell therapies and red light, and so far so good. Again, we can see that here when they're using this, they do some culture expansion of things. You know, questionable with the FDA. Well, I shouldn't say questionable. The FDA says you can't really do it, but whatever, you know, some of us are here and there. All right, so basically exosomes. We know that it can increase exosome production. Okay, stem cell pathways, signaling pathways, etc. And now it also has an effect on the extracellular matrix. I think that's the next subject we're going to start seeing a lot of research on, because it's so important, you know, where the cells hang out, the neighborhood that they hang out in. And so basically what it can do is it can stimulate collagen synthesis, matrix metalloproteases. Those are really, you know, we used to think, you know, everybody was doing A2M to get rid of them all. Well, you know, you have some matrix metalloproteases that are very important and some that are not. But some matrix metalloproteases are what release stem cells from the bone marrow and things like that. And then you have elastic fiber enhancements. You know, that's your elastin and things like that, and fibronectin. So it can have an effect on the extracellular matrix. And those of you that are using allergenic products like Wharton's jelly, you might want to consider photoactivating that Wharton's jelly before you use it in the patient. Okay, so you can see here how, for instance, violet and green can have a very good effect on the extracellular matrix. I'll get into that in just a little bit. So again, we see how the wound healing is affected by the extracellular matrix. That's paramount to getting a wound to heal, the extracellular matrix. And this is a nice slide that you can look at your leisure that kind of gives you the ideas, primary intervention, then some cellular change that we're getting in wound healing. Then we have the cascade, and then we have the final wound healing. And again, all beginning with that photoactivation of the mitochondria and things like that. Now, another view of the wound healing, same thing. I think I showed you a similar slide previously. Now, let's look at it in the brain, photobiomodulation in the brain. We know that this can have some profound effects. So what does it do? Angiogenesis, SOD, anti-inflammatory, cell migration, neuron health, increased cerebral blood flow, neurogenesis, so many different things that you can see here. And it also helps influence neurotransmitters such as dopamine, serotonin, and glutamate. All right, so basically in stroke, I just gave you some ideas of what we're doing. I do some work down in Brazil. I have a lot of good friends down there. We do a lot of research together, publish papers together. And we're doing some work on spinal cord injuries down there. And I'll show you, I think, the next two slides. We've had pretty decent results. We're injecting it intrathecally, and we're using red light laser, getting some good results that we're going to hopefully publish probably in 26. But you can see here how this works. And again, photomodulation decreases M1 macrophages. We heard about that a little earlier, okay? You want, M1s are the ones that are going in there when you have an injury, but you want to get rid of those guys. You want the M2s to start causing regeneration. And that's an important aspect of things. Senescence, cellular senescence. Remember, the zombie cells. Okay, what this can do is it can actually reverse it. You know, we're really, I'm giving a talk the end of this month in Geneva on cell senescence and regenerative therapies. And I think some of the newest thinking now is, yeah, senolytic agents are good, but the best thing is maybe if we can bring those cells back, get rid of the senescence in them, and make them healthy cells again. And that's what I think the real trend is going to be. And photomodulation can help reverse some of that cellular senescence in age MSCs. You can see here's an age senescent cell. You see how it's irregular. You see how now it's getting treated, and all of a sudden it's becoming a little more regular, a little more round and things like that. I'm not sure if we're going to get right to here, but that's the idea. But photomodulation is certainly a step in the right direction there, okay? And photodynamic therapy is another thing I want to talk about a little bit. So basically we have a photosensitizer, and I'll go into that shortly. It produces a lot of reactive oxygen species, so it can get rid of bacteria, it can get rid of, you know, the virus, and also cancer, very anti-cancer, okay? So basically, I have a picture that shows this slide a little better, I think, right here. So basically, you'll inject something intravenously, and then it basically becomes photosensitized with certain kind of light, And then what it does is it goes to the various cells, produces a lot of reactive oxygen species, and it selectively goes after cancer cells and things like that. Now, I've kind of developed a machine that we do this with, and I named this technique extracorporeal photoactivation therapy, because it's not photobiomodulation, because it's compounds. And what compounds are we doing? Methylene blue, glutathione, resveratrol, curcumin, NAD, EGCG, and berberine. Berberine is the only one that's an oral compound. Everything else we give intravenously in my office. And it helps with their bioavailability, and it makes them tolerate it. So this is the machine we use. We use it for some of our autologous and allergenic products, but now we're using it for various IV products also. So methylene blue and photobiomodulation. Okay, so basically it can be red or infrared, and basically it acts as a photosensitizer. You can use this on many patients with cancers, but it's also extremely good for just making you think better, clearer. People today, they said, man, I really can think much better now. Just remember, can't give it if they're taking antidepressants, because you get something called a serotonin syndrome if you're not careful. But the other thing about methylene blue, and this is why I think it gives you such good mental thing, it can act as a transporter in the electron transport chain. It can actually bypass 1, 2, and 3 and go right straight to 4. And especially if you have some of your chain not working as good, that can make a big difference. So that's why we like this for basically increasing your mentation and things like that. And again, this is where we see it work over there, and basically with Cytochrome C Oxidase. And you can show here, it's promised as an antiviral medication, but this shows you a little better. Red light. Now we do is we photoactivate that methylene blue prior to giving it to the patient intravenously. What we used to do is give them methylene blue, then put them on a red light bed. But now we do both. We put them on the red light bed, but we give them photoactivated methylene blue before it even gets to them. And you can see here, this is how it goes with the mitochondria and things like that. All right, that's the curve I've been talking about, the hermetic curve. You want to always be around here. Too little, it's not going to work. Too much, bad things. Okay, so there you go. So important aspects about methylene blue. And methylene blue can diminish nitric oxide production if you're not careful. So you got to be a little careful. Too much, again, that hermetic curve can be important. So bottom line is also, if you're going to use methylene blue, maybe take a nitric oxide product anyway. Now, again, it's the mitochondria that it's really affecting. Now, a good example is right here that you can read more about this, okay? Curcumin, another favorite of mine, okay? Another photosensitized compound. And basically, we use it with violet light, and it can be good for viruses. We use it anytime we have Lyme's or something like that, cancers, very anti-cancer, et cetera. It stimulates the NRF2 pathway, that major pathway that reduces inflammation in the body. It's so important. And again, we use violet light to stimulate that. And here you can see the various things that it does to these organisms. Okay, and then it has potential antiviral effects, like I say. And these are the various pathways that it can help stimulate, among others. Okay, NAD is a photocatalyst. We can cut the time down it takes us to do NAD by 60 to 70% intravenously. We photoactivate it with blue light, okay? We have the patient take TMG, trimethylglycine, and we have them continually sip on coffee. And I can promise you, you'll diminish the time by about 70% it takes to do intravenous NAD. So we know the importance of NAD, and I'm still a believer in NAD. I'm not convinced by the Nigen, NR. It's a lot more money, 3 times more expensive. I don't think it works any better. I was just at a meeting last week where I was talking, and there was someone talking, saying that. All right, so basically some green light, you can see some of the effects there. Okay, so it can have angiogenesis, brain-derived neurogenic factor, circadian rhythm, mitochondrial protection. So green light is a good thing also. And you can see here what it can do. And then red light, we talked about that, but look at all the things it does. I mean, if I have only one choice of one light, that's the light I'm going to use, red light. There you go. Many tasks of red light, anti-apoptosis, anti-inflammation, et cetera. And photomodulation and arthritis, I think it can help. Certainly we use it on all our patients post-injection or anything like that, okay? And blue light is an interesting one also. We used to think, well, not much to it. Now when I say blue, I'd probably also say violet also. And you can see here, blue light stimulates exosomes from MSCs, so an important thing to do. Another thing there. Ultraviolet light can stimulate heat shock proteins, especially ultraviolet A. Heat shock proteins allow these proteins to fold in the cell properly. And you can see that here. And then what it does, here's a heat shock protein. It's like a little chaperone. Make sure that that protein folds properly. And then it also stimulates what we call P53. P53 is basically the tumor suppressor gene. We have only one copy. That's why we're prone to cancer. Elephants have 10 copies. That's why they almost never get cancer. And we can have this here. We have something called negalase. And this basically, I'll show you what it does here. It basically disables your mitochondria. So I'm just about out of time. So I think what we'll do is show you one more slide or two more slides. And that's it. So basically, negalase can be disabled by photomodulation. And it's been my pleasure and honor to talk to all you guys. Thank you so much.